So hello everyone, welcome to another CAP seminar. Today we have an old member of the CAP family, María Ángeles Lezcano. And she is now a researcher, a principal investigator of the Aquatic Ecology and Biotechnology Group. She's working there since the 2022 in the IMDEA Water Institute of Madrid. Her work is focused on the microbial ecology and the aquatic and terrestrial ecosystem, including extreme environments. But also she is involved in the development of nature-based solutions for the removal of contaminants from the environment, such as bioremediation. But she worked here for 4 years, almost 5, doing a postdoc, doing mostly paleobiology of extreme environment using the analysis of biomolecules such as DNA, proteins, and lipids to understand past microbial life. Sorry. And now maybe Víctor, you can introduce her better than me.
Yo solo quiero decir, además lo voy a decir en español, simplemente darle las gracias a IMDEA, como lo llamamos nosotros, por venir al centro a darnos un seminario de lo que está haciendo ahora. La verdad es un orgullo ver cómo personas que han pasado por el centro se han terminado de formar aquí y ahora tienen y lideran sus propios grupos en otras instituciones, pues es un gusto y IMDEA yo creo que se lo merece, está muy bien. Vino con un postdoc de la Comunidad de Madrid, luego con un Juan de la Cierva, y se ha ganado con sus méritos propios el estar ahí y liderar su grupo en el IMDEA. Nada, gracias por venir.
Muchas gracias, Víctor. No sé si se me escucha, creo que no. Pero no con el micro, ¿no? ¿Hola? ¿Hola? Uf, tiene que estar más arriba, creo.
En el Zoom escuchamos muy bien.
¿Está bien? Vale, así. Okay. Thank you very much. I mean, good morning to everyone. Thank you very much for inviting me to come here. Okay, to inviting me to come here. It's a pleasure to come back. When I did my postdoc research I think it's not working very well. Okay, I will move forward. So it's a pleasure to come back and to see all the faces that some of them are unknown for me, but most of them I can remember them. So today I'm coming here to explain one of the studies that I started when I was doing my postdoc here at CAP. This study is related with the microbiology in sediments from a lake that is located in the Andes. But before starting with it, I would like to introduce myself a bit for those that don't know me. In general, during my career, I'm working in microbial ecology, which means the interaction between microorganisms and the interaction of microorganisms with the environment. So I was working in different types of environments. I started with research doing my master thesis. My master thesis was related with toxic cyanobacteria. This toxic cyanobacteria is a type of bacteria that grows in different ecosystems, in terrestrial, in water. But at that time, I was focused just in those that were in the water. And this type of organisms may grow massively in water reservoirs. Probably you have seen that in summer, in lakes or in ponds or whatever. And they can produce toxins that are a problem for human and animal health. So during my master thesis, I was studying the chemical diversity of these cyanotoxins. Then I did my PhD thesis related with one nature-based solution, like the use of bacteria to biologically remove this toxin from the water. In fact, I isolated some bacteria that was able to do this work, and I characterized the genes, the degradation efficiency. And thanks to this work, we have an international patent with a method to remove this toxin from the water. Then in 2018, I came to Centro de Astrobiología to study the microbial ecology of different extreme environments from Atacama or from Antarctica. And finally, in 2022, I came back again to IMDEA Water, which is a center that is in Alcalá de Henares in Madrid. I am one of the PIs of the Aquatic Ecology and Biotechnology group, and our objective is to understand how different environmental stressors, such as temperature or pH or climate change, whatever, contaminants, may affect to different organisms in the aquatic ecosystem, from microorganisms to zooplankton, macrophytes, macroinvertebrates, and even terrestrial organisms. But today, I'm coming here to talk about one of the studies that I developed here. This study, as I said before, relates the microbiology from one extreme environment. It has been already published in the Microbial journal with the work of different institutions. For instance, the University of Duisburg-Essen in Germany, Kiel University as well in Germany, and also the SETI Institute in United States of America. So I will just give you a brief summary of this work, but if you want to go into details, just please, you can download this article, and if you want to see it, you can do it. So one of the main points in astrobiology is to understand the limits of life on Earth, and this is mainly to understand how different microorganisms in extreme environments can deal with these conditions. For instance, low temperature or high pH or low pH. And these extreme environments on Earth has some analogies with other planetary settings in other beyond Earth. So understanding how these microorganisms can adapt to this condition here may understand how could possible life emerge in other planetary environments. One of these interesting extreme environments on Earth is the Altiplano Plateau. This is a high altitude plain that is located between Chile, Peru, Argentina, and Bolivia. It's a volcanic region with many lakes there with different extreme geochemistry. And one of the lakes that pay our attention because of these extreme conditions is the Laguna Lejía or Lejía Lake. This Lejía Lake it's located at 4,300 meters above the sea level, so it's a high altitude lake. It has a pH about 8.5, so it's very alkaline. Has a lot of salinity, and because of the low precipitation and high evaporation rates of this area, this lake, but also other lakes from this region, are desiccating over the last years. So it means that the sediments from this lake are exposed to the atmospheric conditions, which means high UV radiation, to desiccation, to temperature fluctuations. So this is very interesting from an astrobiological point of view. And within this aquatic ecosystem, one of the most interesting settings is the sediment outcrop that you can see in this picture. This is a terrace which organisms that has been inhabiting this place has been in the forefront of different water fluctuations of the lake. They has been exposed to desiccation and to temperature fluctuation as well. And it may serve the microorganisms that live there may serve us to understand the past ecological conditions of the lake, and also the geochemistry can give us some clues about what happened in the lake in the past. So this is our target for this study, and we have several hypotheses for this study as well. In this terrace, we think that we can find a mixture of microbial remains, but also microorganisms that are still living there. Because despite this terrace is from the late Pleistocene, we cannot isolate this environment from nothing. So there are microorganisms everywhere that can go into the sediment. So we think that this could be a mixture of different extinct but also extant organisms. The disconnection of the water level of the lake also may have caused a new ecological niche for these microorganisms to live there. These microorganisms also may have some adaptations that we need to know what are they to live in this environment. And as it is located in a very remote location, we think that we will find some novel microorganisms. So the objective of this study is to characterize the mineralogical and geochemical environment of this lake, and also to radiocarbon date these sediments to know exactly how old they are. We will also like to explore the microbial composition and the relative abundance of them. This microbial composition, we will define the bacterial community and the archaeal community. And then we will want also to estimate if these microorganisms are still living. So we will do it by calculating in silico replication rate of the genome. I will explain later on what does it mean. So in 2018, in November of 2018, we were, well, not me, but my colleagues were to sample these terraces in Laguna Lejía. And the first thing that they did is to remove the outer sediment of the profile in order to be able to remove the probably organisms that are modern organisms that could obscure the analysis of our sediments. So they removed these sediments, and they were collecting 3 different layers based on different color and texture across the vertical profile of the sediment. So this vertical profile is just 1 meter. They collected just 1 meter high of these terraces. Then they came back with the sediment, and we did several analyses to give answers to our questions. One of these analyses, the two radiocarbon date, to analyze the radiocarbon of this organic matter to understand how old are these sediments. We analyze also the mineralogy, the geochemistry, lipid biomarkers, and DNA by doing some metagenomics. I'm going to drink a little bit of water. So what radiocarbon dating gives us to know from these sediments? So these analyses tell us that our samples date back to 11,000 years before present. Well, depending on the sediment layer, they gave us different dates. But in general, they dates back to the late Pleistocene, so it's 11,000 years before present. And from the sample E to the sample F, which is in the uppermost part of the terrace, we are considering 700 years. So our questions is what is happening in the lake during these 700 years. So the mineralogy and the geochemistry give us some clues. If you can see on the left side, the mineralogy shown different alternance of minerals across the sediment profile. One of the most abundant minerals is magnesium calcite, which is the red one, but also some alternance with different feldspars. This pattern can be also observed in the analysis of different water-soluble elements. In the graph in the middle, we can see the major elements in the sediments. Most of the elements are chlorine, or the most concentrated are chlorine, sodium, but sulfur as well. And the less concentrated, but that deserves to mention, we found some arsenic, lithium, boron, etc. As you can see here, there is a similar pattern with the mineralogy and the water-soluble elements, which suggests that there has been several water level fluctuations during these 700 years. In fact, we think that, for instance, layers A, C, and E, which has higher concentration of ions and even higher concentration of magnesium calcite, may be related with periods where the water column of the lake was lower, and then there were higher concentration of the ions that give to a sedimentation of this precipitation of these elements. But also beyond this pattern of alternating desiccation and water recharge events in the lake, the presence of sulfur can give us also information that the volcanic region is also influencing this lake, this environment in general, and also the presence of different arsenic and other components such as lithium. It also may be related with the climatic region in the Altiplano. We also measured the total organic carbon and the total nitrogen. We found the similar pattern, as you can see. So it also reinforces the idea that there were some desiccation and water recharge events in the lake, but also we have very, very low concentration of total organic carbon, which means that this environment is oligotrophic. So how these organisms live there. So we don't know exactly right now. So I will tell you later on. So considering that there were water fluctuations along these 700 years, what happened with the biology that was there? So we analyze, as I said before, DNA and lipids. But lipids can give us information, very interesting information, because lipids, compared to DNA, they have been preserved in the geological record for the durability of these lipids are higher than the DNA, but they don't give so much information in terms of a specific name with a surname of a microorganism, but it can tell us if there were aquatic plants or if there were some microorganisms or not. I mean, just in general terms. So the information that give us the lipid composition is that we have, well, sorry, I forgot to say that even these lipids are very well preserved in the environment. We usually distinguish between 3 lipid families, which are the non-polar fraction, the polar fraction, and the acidic fraction. And each family has, for instance, the acidic and the polar fraction is the lipid with different carboxylic or hydroxylic groups that are more prone to decay over time, which means that if we found them in our environment, it means that it's well preserved. Because if there has been any kind of chemical degradation, most of our lipids would be the alkanes, not the non-polar fraction. So the higher proportion that we found here in all the sediments of higher proportion of acidic and polar fraction compared to the non-polar one, it means that our biomass is very well preserved in this environment. Also, our analysis of the lipids also give us information that we have some aquatic and terrestrial plant remnants. We found some esterols that were related with terrestrial plants and aquatic plants. For instance, campesterol, cystimaxterol, that I'm not going to go into details, but just for you to know. But let's go now for the microbial community. So to study the microbial community, as I said, we analyze the DNA. So the way we analyze it is the technique of metagenomics, which we don't need to culture them in the lab, but just we take the sediments from the environment, and then we extract the DNA, and we try to reconstruct the pieces of the DNA to make the complete genome of every microorganism that are in the sediment. Okay, so with the complete genome, we have 2 types of information. Who they are, I mean, if they are with surnames, with numbers, with names and surnames, so with up to the species level, but also the potential or the function of these microorganisms in this environment. So let's start just to know who they are. So most of the organisms here are from the phylum Actinobacteria, Proteobacteria, Chloroflexota, and Paterstibacteria. I'm not going to tell more names because it's really boring. And this community has been compared with the community that was in the wet sediments of the lake. We didn't did this work, but we compare it with other studies that were performed before. So some people, some researchers went there and they went to the lake and then took the samples from the sediments, so wet sediments, and they found a completely different community, mainly comprised by Bacteroidetes, Firmicutes, and Proteobacteria. So it's different. So we can say that the desiccation of the lake provides a new ecological niche for these microorganisms. So while the lake was desiccating, the microorganisms were anyway living in the terrace and then trying to well, they were living in the terrace, and that's all. But how can we say about these organisms? Because what I show you is just at the phylum level, which is if we compare us, like the phylum Chordata, which is the human beings, with arthropods. The comparison is not very well. So we have to go more into details to go to the species level to understand if the community, the microbial community that is in these sediments are new or they aren't. And we do it by doing some phylogenetic analysis of the bacteria and the archaeal community. So we did it, we were able to reconstruct 572 bacterial genomes, which is a huge amount of bacterial genomes, and 19 archaeal genomes as well. With this information, we realized if we go to the graphs on the right, we realize that, for instance, if we go here, we can see that 99.7% of these bacteria was unclassified to the species level, which means that we have a lot to know about the organisms that are living there. They are not classified in public databases, so they are completely new. And the same or similar happens to the archaea. 74% of the archaea were not classified up to the species level. So in fact, if we compare this percentage of novelty with other studies, for instance, in the Arctic Ocean, we found that in the Arctic Ocean, 33% of that species were completely novel. And here we have 98.8% of species completely novel. So there is a huge amount of work to do in this environment. Probably this could be also explained that the ability to isolate bacteria from this environment in the laboratory is very, very difficult. And to identify new species, we have to do it in that way, so to isolate from the environment. But also, this is a location, a remote location with very limited access and no studies in this area. So we found an amount of organisms that are really, really novel, with probably a lot of adaptations, new adaptations in this environment. Which are these adaptations? So as I said, with this genome, we could infer the metabolisms that these organisms were doing here. So on the left side, I lost the point, I think. Well, on the left side, you can see the phylum of the bacteria and the archaea. They are the names up to the phylum level. And in the columns, you will find the metabolisms. I try to make it easier on the bottom of the table to give you information of the element involved in this kind of metabolisms. For instance, the carbon, nitrogen, sulfur, arsenic, and many others that are there, like copper or halogen compounds. And in general, if we see this picture, what we can see is that most of the metabolisms that can be performed by this bacterium are related with sulfur. And as I said before, there were a lot of sulfur concentration because it's a volcanic region, so it makes sense. But also a very interesting thing is that a lot of, here is the point. a lot of microorganisms can do carbon monoxide oxidation. As I said, there were very low concentration of total organic carbon. So how could they live here if there is not almost organic matter? So they oxidized the carbon monoxide. And in fact, it was already described that the oxidation of this molecule has been described in oligotrophic environments. So it makes sense that we could find something like this here. We have also a lot of organisms, in fact, all of them, except cyanobacteria, that has resistance to arsenic, and also they can metabolize them. And this is quite interesting because we found also arsenic in the lake. So they are adapted to this environment, so it's not toxic to them. It's also very interesting that we found halogen metabolisms. This is very, very interesting from a biotechnological point of view, because compounds with halogen compounds are very resistant in the environment. So these microorganisms can actually metabolize them. So they can remove it from the environment. And, So in general, having all this kind of adaptation in this environment in very distant bacteria and also archaea, it seems quite strange. So we have to think what happened there, why they have so many adaptations in the very distant bacteria. So what it suggests us is that there could be some possible horizontal gene transfer. So they give us, if you have next to a cell, you can give the gene that is involved in the metabolism or the arsenic or something like that to be able to live in this environment. It's also very interesting. I'm not going to go more into details, but I would like to say that there are, with this possible horizontal gene transfer, there were a lot of new phyla with new metabolisms that were not described before. So, for instance, the phylum Methylovirabillota has the possibility to do thiosulfate oxidation that was not described before. We found some genes to do the Calvin cycle, which is the photosynthesis as we know in plants, in Nanoarchaeota. We don't know exactly why we found it, but maybe they could do it because we found these genes that predicted the enzymes to do this kind of metabolisms. And in general, many other metabolisms in these environments. But are they living there, or they are extant? So for answer this question, what we did is to doing a calculation with the genome. So when a cell is replicating, they need to replicate the DNA as well. So if we calculate the number of copies at the initial of replication compared to the terminus of the replication, we will have a ratio. If this ratio is above 1, it means, yes, it's above 1, it means that this bacteria is replicating in the environment. We have here a threshold of 1, just to make it easier. And we can see that most of the bacteria is above 1. Many of them are close to 1, but most of them are above 1, which means that these bacterium are replicating, they are not extinct, they are alive. And we found that in the layer E, we found a lot of replication. We don't know how to answer this question. It's maybe for other people as well to investigate that, but we said our results suggest that at this layer, there were a lot of sulfur, and maybe bacteria that is adapted to this environment can grow better in this layer than in the others. So our conclusions is that our results in the community structure, the geochemistry and the mineralogy, suggest that there were water level fluctuation in the lake, that the lipid biomarkers also gave us information that there were terrestrial and aquatic plants from the past, when the water level reached the terrace of the lake, that the broad genetic and metabolic diversity of these microorganisms are adapted to this extreme environment. Especially, they can do oxidation of carbon monoxide and also metabolism related with the sulfur, the nitrogen, and halogenated compounds. And that we found a high novel microorganisms in this environment, and even many of them with undescribed metabolic activities. And so it expands our knowledge of microorganisms that can do some specific metabolisms in extreme environments. But I'm so sorry, I'm not going to finish because I'm coming from IMDEA, and I would like just to give you a brief overview of what I'm doing right now at IMDEA Water. IMDEA Water is a research institute that is located in Alcalá de Henares, Madrid. You know them because we are close by. And this center, it's one of the 7 centers from the Comunidad de Madrid. This is just related with water research. And we are almost 85 people right now in the institution. And we have 2 directors now, Irene de Bustamante and Rafaela Mefe. We are 5 research groups and another group that gives support with analytical services. One of the group is the soil and water quality, bioelectrogenesis. In fact, the PI of the bioelectrogenesis group is Abraham Esteve, who was also a researcher here at CAP long time ago. The aquatic ecology and biotechnology group, in which I am, membrane technology, and economic and institutional analysis. Just to give a brief overview of our group, we are more or less 14 people right now. We are several PIs. I am one of them. And we have several active projects, some European, some regional, and some national. But now I'm going to just to give some words about one of the projects that I am leading together with Pablo Urrutia. As I said before coming to CAP, I was working with toxic cyanobacteria. It's a big problem in water reservoirs, especially if they are intended for drinking or for recreational activities, because these toxic cyanobacteria produce toxins that are dissolved in the water, very difficult to degrade, and they can be drinked by tap water if the water treatment plants don't do the correct work, or if you go just for swimming and you drink it, and so on. So there are many different cyanotoxins that produce problems to the liver. They are also neurotoxins. And so it's a very health and for humans and so on for animals. It's a health problem. And then we were granted by a project in which we, as I said before, when I was doing my PhD, I isolated one bacterium that was able to degrade these toxins in a high efficiency. So we are granted by a project that is named Cyanotox, in which we will use this bacterium to try to to remove this cyanotoxin from the water. That can be useful not only for ponds or for swimming or for reservoir or whatever, but also for the industry, for instance, aquaculture, fish farms, aquaponics, or even for drinking water treatment plants. So what I did in the past was to test this bacterium in laboratory conditions, in sterile conditions, but now we are moving forward to make it the application more real. So at IMDEA Water, we have a mesocosm infrastructure in which we mimic different lentic and lotic environments, which means ponds or rivers. And then we will add the bacterium in different conditions, but in general in the Mediterranean climate, which is here at IMDEA in Spain. And also we will move to Sweden in collaboration with the Uppsala University to use as well another mesocosm infrastructure and to test this bacterium under boreal climatic conditions. And just to finish, just if it may be interesting for you for any kind of collaboration, at IMDEA Water, we are very good at different pilot scale experiments. We have membrane technologies. We have bioelectrochemical microbial technologies such as wetlands, mesocosm that mimic aquatic environments, as I said before, green filters with a popular community, being able to evaluate how emerging contaminants can go through the across the soil, and also a greenhouse to make experiments. We have also a very potent laboratory with many infrastructure for analyzing contaminants and other elements. For instance, we have different liquid and gas chromatography coupled to mass spectrometry, ICP-EMS, and different analytical instrumentation to measure micro and nanoplastics. And that's all. Thank you for your attention. And if you have any question, feel free to ask.
Thank you so much, María Ángeles, for sharing your work with us. Anyone has a question?
Hi, María Ángeles. Thank you for your research and your work. The paper you told is an amazing work and a huge effort. So congratulations. And I have one question about the microbial composition of communities during all the 700 years. Did you find, maybe I missed it, did you find any changes in the microbial composition in the wet or dry seasons of these years?
I'm going back. Do you mean in the vertical profile of the terrace or compared to the sediments of the lake? Or...
Yeah, I mean, you told that about the mineralogy and the geomicrobiology, that it is supposed to, the lake to fluctuate and to lose water. And is it reflected to the microbial communities? I mean, maybe in the wet periods, actinobacteria has a higher relative abundance or something similar. Did you do that work? Or...
Yes, I mean, I think it's a very interesting question. We did it. We compared the microbial community across the different layers, but we didn't find so much differences, except, I didn't put this image here, but except the D layer, which was a little bit different, but with not so much difference between them, which we explained that during the desiccation of the lake, it was just a completely different environment for microorganisms, and what so the microorganisms that inhabited this environment was more or less all the same.
The same, okay.
Yes, but anyway, we found traces of DNA that we are sure that come from moments in the lake that were more wet. I mean, for instance, here in this, I'm sorry. For instance, we found cyanobacteria that has the Calvin boson, well, that do the photosynthesis, and then it means that it comes from a period that was wet. I mean, anyway, cyanobacteria can be in very arid environments as here, but it's a lake, and so in fact I didn't put it here, but it was not in the upper layer. It was in the middle of the layer, so it comes from the past. I mean, there are DNA remains from the past.
Okay.
So,
thank you.
You're welcome.
Any other question?
Thank you very much for the clear talk. My phone almost died. I was wondering. I was wondering in this. I think it's the next slide. That you showed that there's a difference for the E level, no, and that's due to the sulfur. So there was some kind of volcanic activity. No, I think in the one that you showed before.
But you mean this one?
No, the one at the end. Next, that one, no?
Okay.
So you said that at the E level, no, at the E
Yes.
substrate.
See.
So there's a difference in the replication, no? So does that mean that they are replicating more or less than for the other levels? Because I didn't understand very well.
They are replicating more.
More.
Yes.
And that's due to the volcanic.
We don't know exactly how to interpret these values, because usually when you find values above 2, it means that there are different initial replication places within the genome. So it's very difficult to interpret this value, but we think that maybe it could be related with the possibility that there are higher concentration of sulfate, that these bacteria can use it for, maybe it's better for them to grow in this layer than in the rest. But this is just what the graph suggests us. But of course, yes.
Yeah, let me think about it.
1 second.
I don't remember, but there were some more sulfate reducers in the samples. I don't remember that. It's because it could be related with the accumulation of sulfate, or I don't know if it is sulfate reducers or sulfur oxidizers. I don't remember if there was a difference in that layer.
Yeah, sulfur, there are different oxidation and reduction of different species of the sulfur. And here I didn't put in which layer they are found in higher concentration, but they were widespread in all the sediment layers. So maybe somehow they are more active. We don't know exactly. What we can show is our data. We interpret that maybe they are better in this layer for maybe a reason that we didn't exactly measure with our data. Maybe we have to do any more analysis, not just geochemical or mineralogical. So maybe more, just to understand why they could be more active. Because actually genes for doing oxidation of sulfur or species or reduction of sulfur species are across the sediment profile. There are different phyla that do it. So we just could say that they are replicating more.
Any more question? I don't know if you want to add something else or. Any more question in the room? I don't know if someone have questions in the Zoom, but okay.
In your facilities that show us at the end, in these mesocosms facilities do you have a different type of sensors that can monitor parameters in real time? For example, I don't know, how pH change, how the ionic strength, etc.
We are going to put it because of our experiments in the Sinotox and other projects. We need to monitor in real time the temperature, the salinity, and the pH. But we don't have it now in this picture. But we are going to put it because it's necessary for that. Yes. So we'll do it.
No, but I mean that controlling everything in an open system, it's a mesocosm, but somehow it's an open system because you have everything exposed to the sun, to temperature, a
yes.
lot of things. It's rather complicated, but
yeah. But this is what makes interesting, because
yeah.
Otherwise it's the laboratory condition that is not a real condition.
Yeah, but also you have to be more precise
yes.
in following all the parameters, because otherwise the conclusions.
Yeah, it's many times it's very difficult to.
It's okay. It's a good. Thanks.
Can I ask another question? Sorry. So it's regarding the measurements you showed for the isotopes of carbon and nitrogen. Yeah, so there are a lot of changes, no? So there's a big change in the substrate B and D. So what is that produced by?
You mean in substrate? Oh,
Yeah, there's a decrease, no? Substrates B and D.
B?
For
You
B
mean the
and D.
isotopes or the total organic carbon? You
Ah,
mean
that's the
the
total organic carbon.
No, it's both. I mean, I just stress on the total organic carbon, which is the bars.
Right?
But if you look at the dots, you will find the measures of the isotopes.
Ah, and then that's similar in across all the layers.
Yes, I mean, the pattern is the same as the one in the mineralogy, the one in the geochemistry, I mean, the concentration of the elements. And so we think that it could be related with the water fluctuations. We can explain this with the water fluctuations in the lake.
But the refilling and the drying, drying cycles.
Yes. In fact, it was also described by some geologists in the area, but they described just three water desiccation and recharge events. But with our data, we suggest that there could be 5. So it could be like a period of this desiccation, like desiccation, dilution, and desiccation again.
Nice. Very nice. Thank you.
Yes, there is a question on Zoom by Maripaz. You can mute yourself and go ahead.
Yeah, thank you. Thank you so much. Hi, I've enjoyed your talk very much. Coming back to the enhanced replication at the layer where sulfates are, I wonder if this may be caused by something which we propose for Mars, that sulfates are hydrated. They very easily capture water from the atmosphere and they remain hydrated with 2 or 4 molecules. So one possibility could be that cells that are within that layer have access to water in a very dry environment, whereas other cells that are not so close to salts don't have a water reservoir. Do you think this could be the case?
Yes, sure. I think that it could be completely the case that the humidity in this layer could be higher. In fact, we didn't measure the humidity in situ, so it could be possible that there could be some higher humidity here because of that. And that could explain why these microorganisms could be more metabolically active. So yes, I can buy your explanation.
Great. So I like this environment then as an analog. Thank you.
Well, welcome.
I think we don't have any more questions on Zoom. I just have a very quick question regarding preservation. You said that you identified both extinct and extant communities, right? And they were very different from the ones habiting in the lake. Do you know if the fact that there are still active organisms are changing the signatures that you get from the previous existent organisms, or is it still the same? I mean, you are able to characterize as well as if any biological activity was, finished.
I mean, it's a very interesting question, but very difficult as well to answer.
Sorry.
Yes, as I said, I mean, we are right now working in an environment that we know that is 11,000 years before present. I mean, the organic carbon that is there has dated back to 11,000 years before present, but it's not completely isolated. So there are microorganisms everywhere that colonize everything. In fact, the sediments has pores where the microorganisms can go inside. There is a possibility that, I mean, it's a possibility that there are some humidity. It is the water table of the lake is not right now touching the terrace, but there are some kind of humidity in the environment. So it's impossible to distinguish which is extant and which is extinct. But the only thing that we could say is that we know that some microorganisms are replicating, and we also know that some microorganisms make photosynthesis, but that in in deep layers of the terrace, which means that one day in the past was in the surface. So this is the only clue to understand that these microorganisms were come from the past. But it's really difficult to answer the question and to, yeah, everything is mixture here on Earth.
So thank you very much. I want to ask
welcome.
for an applause for her, and thank you for coming. Oh, another question, sorry. Oh, a last minute question.
Thank you. No, it's just to add to the question you asked. I think, of course, there must be influence from the extant metabolism operating right now on the signature we measure. But I think the clue is to consider that when we date the organic carbon, it is the bulk organic carbon. And in mean, we established that it is from 11,000 or 10,000 years old, mean value or whatever. So there must be material even older and younger. So in mean, we have at least this age of organic matter. So I guess the most of the signature we measure is old, although with influence from younger or actual metabolisms, I would say. That's.
And can we know if the habitat was similar to the one that we have in the lake now, or is very different from what we had? Because she said that it was very different, ecosystem. So I'm not sure if.
That I don't know, but I would say that it is more similar to the past, because if we compare with samples and analysis from nowadays in the lake, in the existing lake, they seem to be quite different. So I would say it changed very much because of the climatic conditions in the past, and then the metabolisms and organisms adapted to that, and they somehow remained stopped, although there are some metabolisms still going on, but I guess in a very lower level. That's how I read it.
Thank you very much. And sorry for the applausus interruptus, and now. Thank you.
Thank you.
No te olvides tampoco de las cianobacterias. Yo creo que pueden estar vivas, no pueden estar igual que las de tinto, están tiernas las cianobacterias.
Gracias. Sí,